Nucleic acids. Nucleic acids are polymers called polynucleotides, and the monomers are called as nucleotides. And the component of a nucleotide, it is made up of a nitrogenous base, which is bonded to a pentose sugar, and the pentose sugar is bonded to a phosphate group. So let's draw the structure of a nucleotide. So basically, because it is made up of one of the component is made up of pentose sugar, so pentose sugar is a 5-carbon sugar. So 5-carbon sugar, you have to draw it with the pentagon structure here, with oxygen on top of here, and then it is bonded to a base. Okay, so this is the 5-carbon sugar or a pentose. So this sugar can either be ribose or deoxyribose. It depends on the type of polynucleotides, either DNA or RNA. And then here is the sugar bonded to a phosphate group. So if we were to number the carbon, this is carbon number 1, carbon number 2, number 3, number 4, and number 5. So carbon number 5 of the pentose sugar is bonded to a phosphate group. Carbon number 1 is bonded to a base. So the base can either be adenine, guanine, cytosine, or thymine. So this is if the polynucleotide is DNA. So if the polynucleotide is RNA, then the base is adenine, guanine, cytosine, or uracil. So that is the basis for RNA. And then it says here the portion of a nucleotide without the phosphate group is called as nucleosides. So this portion here, where you have the pentose sugar bonded to a base, this region here is called as nucleoside without the phosphate group. In this slide, we can see the structure of a polynucleotide. So this polynucleotide, it consists of several nucleotides that are joined together through a covalent bond. We call it as phosphodiester bond. So this is the phosphodiester bond that connects between adjacent nucleotides. Okay. Alright. So as you can see, a polynucleotide, it has specific direction. So a polynucleotide has to start with a 5' end and ends with a 3' end. So the number is basically indicating the number on the of the carbon of the pentose sugar. Okay, so carbon number 1, carbon number 2, number 3, number 4, number 5. So carbon number 5 is bonded to a phosphate group of the pentose sugar of the pentose sugar. Okay. So downstream, the last nucleotides will have a free 3' end that has a hydroxyl group. Okay, so if this polynucleotide to be added a new nucleotide to make the polynucleotide longer, so the next nucleotide will be added to the 3' end. Okay, to the 3' end of the 5-carbon sugar. Alright, so therefore the direction of the polynucleotide always goes from 5' to 3' end. Okay, so and as you can see here, it says here sugar phosphate backbone on the blue background. Okay, so the polynucleotide has a sugar phosphate backbone, which consists of repeating unit of phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. So this is what we call it as phosphate sugar backbone. And protruding from the sugar phosphate backbone is the you have the bases. Okay. So the bases can be adenine, guanine, thymine, cytosine for DNA, or replace the thymine with uracil for RNA. Okay, so this is the structure of a nucleotide. So you have the 5 carbon sugar or the pentose sugar. So carbon number 1, carbon number 2, number 3, number 4, number 5. Carbon number 5 is connected to or bonded to a phosphate group. And as you notice, the phosphate group is negatively charged. Okay, it is negatively charged. And the portion that consists of only the sugar and also the base, we call it as the nucleosides. Okay. Nitrogenous bases. Two families of nitrogenous bases are pyrimidines and the other one is purines. Pyrimidines consists of three different types of bases, which is cytosine, thymine, and uracil. So pyrimidine basically is a single six-membered ring. Okay, so as you can see, cytosine, thymine, and uracil, it is single six-membered ring. Okay, 1 2 3 4 5 6. Okay, single six-membered ring. And as for purine, which is consists of adenine and guanine, it is a six-membered ring fused to a five-membered ring. Six-membered ring fused with to a five-membered ring. So adenine and guanine. Phosphodiester bond. Phosphodiester bond is a type of covalent bond between adjacent nucleotides. As you can see here in this diagram is two nucleotides that are bonded to one another through the formation of phosphodiester bond. So you have to know exactly where to label the phosphodiester bond. It is between carbon number 3, okay, carbon number 3 of the first nucleotide to the phosphate group, okay, phosphate group of the next nucleotide, which is this phosphate group is bonded to the 5 prime carbon, okay, the 5 prime carbon of the next nucleotide. So the bond is between carbon number 3 and also carbon number 5 here. Okay, so that is the phosphodiester bond. So phosphodiester bond is formed between the hydroxy group on the 5 prime carbon of one nucleotide here, okay, this one here, to the phosphate group on the 5 prime carbon of the next nucleotide. Okay, so the chain of the nucleotide has a 5 prime to 3 prime orientation. So a DNA strand or RNA strand, okay, the orientation always start from 5 prime to 3 prime and okay, so from 5 prime to 3 prime and so the backbone is negatively charged due to a negative charge on each phosphate. As you can see, the phosphate has negative charge. So this causes the backbone of a DNA or RNA to be negatively charged. Hence the name is, for example, DNA is deoxyribonucleic acids or RNA is ribonucleic acids. Okay. There are two types of nucleic acids. One is deoxyribonucleic acids or DNA, ribonucleic acids or RNA. Okay, so you have a cell here, for example, and inside the cell, there's a nucleus. Inside the nucleus is where you can find the DNA. So DNA is basically a very large molecule that stores genetic information. And all this genetic information is stored in the form of genes. Okay, and this DNA can undergo DNA replication. Okay, to replicate new DNA in a new cells. Okay, in a new cell. Okay, so DNA can also be found in other organelles, which is in chloroplast and also mitochondria. Okay, but not as much as in the nucleus. So DNA directs the synthesis of RNA. So RNA, there's messenger RNA or mRNA, and also there's tRNA or transfer RNA, and the other one is rRNA or ribosomal RNA. And all these RNA involved during protein synthesis. Okay. In DNA, the pentose sugar is deoxyribose, and RNA, the pentose sugar is ribose. As you can see here, you can see the difference in the structure of deoxyribose in RNA and also ribose in RNA. So the difference is that at carbon number 2 of deoxyribose, there's hydrogen, but at carbon number 2 of ribose, there's hydroxyl group. Okay, the rest is the same. DNA double helix. DNA has two polynucleotides strands spiraling or coiled together around an imaginary axis, forming a double helix structure. As you can see here, a DNA molecule consists of two strands that are coiled together, and these two strands are held by hydrogen bonds between the two between bases, okay, between bases along the polynucleotide chain. So the two backbones in the DNA double helix run in opposite direction, which is in the direction of 5' to 3' direction, and this arrangement we call it as anti-parallel. So as you can see, if I were to straighten this helical DNA into a straight chain, okay, so one strand will start with 5' end and ends with a 3'. So 5' has to pair with a 3', 3' has to pair with a 5', and the direction of these two strands, it runs in opposite direction or anti-parallel. So 5' will run in the 5 to 3' direction. On the second strand, it also runs in the 5 to 3' direction. So these two strands runs in the opposite direction. We call it as anti-parallel. Okay, anti-parallel. So the two strands has to run from 5 to 3, 5 to 3. So anti-parallel. So the two strands, okay, the blue strands here is the sugar phosphate backbone, repeating unit of phosphate sugar, and protruding from the backbone are the bases. Okay, so let's say one strand, a base adenine will pair with thymine, guanine will pair with cytosine, and the pairing between the bases are held by hydrogen bonds, adenine and thymine, guanine with cytosine. And the hydrogen bonds between adenine, there's formation of two hydrogen bonds between adenine and thymine, and between guanine and cytosine there's three hydrogen bonds. So these hydrogen bonds will hold the two DNA strands together. Okay. DNA double helix. One DNA molecule includes many genes. In your nucleus, the large molecule of DNA will contain many genes as it contains or stores genetic information, and each of the genes will code for the synthesis of specific protein. The nitrogenous bases in DNA pairs up and form hydrogen bonds. Adenine will always pair with thymine, guanine will always pair with cytosine, and the pairing between adenine and thymine, two hydrogen bonds are involved, and as for guanine and cytosine, three hydrogen bonds will involve, and this is what we call it as complementary base pairing in the DNA molecule, and the two DNA molecules are held together by these hydrogen bonds. So in this slide, we can see the pairing between the bases, between purines and pyrimidines. Purine, as you know, it consists of adenine and guanine. Pyrimidines in DNA consists of thymine and cytosine. So along the DNA strand, adenine will pair with thymine with the formation of two hydrogen bonds, and guanine will pair with cytosine with the formation of three hydrogen bonds. In this slide, we can see the structure of a DNA molecule and the structure of a tRNA molecule or transfer RNA. So DNA molecule is consists of two strands that are coiled together to form a double helix. Okay, and the direction we start with 5 prime and ends with a 3 prime, and the other strand we start with 5 prime and ends with a 3 prime. So this the two strands that run in opposite direction, we call it as anti-parallel, and the two DNA strands are held together by the hydrogen bonds that are formed between the pairing of the bases along the DNA molecule. And then the other molecule here we can see is RNA, and this type is transfer RNA. So this is an RNA that folds into a specific shape, forming tRNA. So basically RNA, it is single strand molecule, but it seems here it is double stranded, but it is actually single strand molecule. In this slide, we can see the difference between RNA and also DNA. RNA, it is single-stranded molecule, and as for DNA, it is double-stranded molecule. And then for RNA, the molecule is much more shorter compared to DNA. It is much more longer, and it is coiled. The two strands are coiled together. Okay, and the arrangement is anti-parallel between the two strands. For RNA, the pentose sugar is ribose, and as for DNA, the pentose sugar is deoxyribose. In RNA, the bases are uracil, adenine, guanine, and also cytosine. And as for DNA, the bases are adenine, guanine, thymine, and cytosine. The difference is in these bases. DNA contains thymine, and as for RNA, the thymine is replaced with uracil. The next difference is for RNA, it is produced in the nucleus, but RNA, due to their small size, they can be found anywhere within the cell because they are involved in protein synthesis. And as you know, the synthesis of protein occurs in the cytoplasm. And then for DNA, found almost entirely in chromosome in the nucleus, and there is a small amount that is also found in mitochondria and also chloroplast. And the next difference for RNA, there is three types, which is mRNA or messenger RNA, transfer RNA or tRNA, ribosomal RNA or rRNA. And then for DNA, there is only one type of DNA, which is the one that carries your genetic information. And then the size of RNA, it is much more smaller. Okay, the size of the RNA is smaller compared to RNA. The size is much more bigger, larger molecular mass. And for RNA, it does not carry genetic information. It does not act as a gene, but it is important during protein synthesis. RNA is really required during protein synthesis. And then for DNA, the nucleotide sequence may encodes for genes. Each gene in DNA is a unit of genetic information. So basically, DNA, it carries your genetic information, where in your DNA it has many genes that code for the synthesis of specific protein, right? So that's the end of this slide. So thank you.